Welcome back to the deep dive. Today we're really aiming for mastery, getting under the skin of connective tissue, understanding that matrix.
Absolutely. It's so much more than just filler material.
Right. It's not passive at all. It's the body's infrastructure, isn't it? The support system, the communication network, defense. It's everything.
It truly is. If you want to grasp how the body holds itself together, how it heals, how it fights off invaders, you have to understand connective tissue, or CT.
So our mission today is exactly that. Mastering the concepts. We're going deep into the matrix architecture, the cells, the fibers, the ground substance, the whole picture.
And it's a great place to start because fundamentally, CT provides that physical matrix connecting all other tissues, epithelium, muscle, nerve-holding organs together.
But it's more than just structure, as you said. The sources really emphasize the metabolic role.
Oh, absolutely critical. That interstitial fluid within the matrix, that's the essential medium for, well, for everything moving between blood and cells.
Nutrients coming in, waste going out.
Exactly. It all has to diffuse through that fluid. It's the vital connection between your circulation and really every single cell.
Which brings us to maybe the most fundamental point about CT. What makes it different from, say, muscle tissue?
The key difference, the main component isn't the cells.
It's what the cells make.
Precisely. It's the extracellular matrix, the ECM. The cells are almost like residents living within this environment they've built themselves.
So mastering CT means mastering the ECM. What's it made of? Let's break it down.
All right, three main things to keep in mind. First, the protein fibers. These give it the physical strength and resilience.
We're talking collagen.
Collagen, yes, for incredible tensile strength, resisting pulling forces. And elastic fibers for that ability to stretch and snap back.
Like ropes and rubber bands, essentially.
That's a perfect analogy. Then second, filling all the space around those fibers, you have the ground substance.
And this isn't just water, right? It sounds much more complex.
Oh, it is. It's a highly complex, hydrated, gel-like material. Think of it as a biochemical soup.
Okay, let's unpack that soup. What are the ingredients?
Right, so the ground substance itself has three major classes of macromolecules. They're all large, negatively charged, and they love water hydrophilic.
Okay.
First, you've got proteoglycans, or PGs. These are huge molecules with a protein core and lots of sugar chains attached. Very good at trapping water. Then there are the glycosaminoglycans, or GAGs. Even larger sugar polymers, like hyaluronan, they're not usually attached to proteins themselves.
And the third type?
The multiadhesive glycoproteins. Think of them as the glue. Molecules like laminin and fibronectin. They have binding sites for the fibers, for the PGs, and importantly for the cells themselves, anchoring everything together.
Wow. So fibers, this complex ground substance gel. What's the third ECM component?
Just the water itself. But it's not free-flowing water. It's bound water held tightly by those hydrophilic GAGs and PGs.
And that bound water is key for, well, everything.
Pretty much. It gives the tissue its turgor, its cushioning ability, and allows for that diffusion of nutrients and waste we talked about. You know, the amazing variety of connective tissues you see, from delicate membranes to tough tendons. It all just comes down to tweaking the recipe.
Changing the proportions of cells, fibers, and this ground substance mix.
Exactly. Different ratios, different types of fibers, different GAGs. That's what creates the diversity.
It makes perfect sense. Okay, let's rewind. Where does all this complex tissue originate during development?
We trace it all back to the embryo, specifically to a tissue called mesenchyme.
Mesenchyme.
Yes, all connective tissues, no exceptions, arise from this embryonic tissue. It mainly develops from the mesoderm, one of the primary germ layers.
What does this early mesenchyme look like under the microscope? Is it already forming fibers?
Not really. It's actually defined by having a very viscous, gel-like ground substance, but surprisingly few collagen fibers at that stage. The real action is in the cells.
The mesenchymal cells themselves.
Right. They're undifferentiated, often spindle-shaped or star-shaped, and if you look at their nucleus, it tells you a lot.
How so?
The nucleus is typically large, oval, and very pale staining, which we call euchromatic. That pale appearance means the DNA is uncoiled, actively being transcribed into RNA. Plus, they usually have very prominent nucleoli, the sites of ribosome production.
So high synthetic activity. They're busy making things, even if they haven't specialized yet.
Exactly. They're gearing up. And here's the really crucial part, connecting the dots. Mesenchyme isn't just the precursor for what we think of as regular connective tissue, like the stuff under your skin.
It gives rise to more specialized types, too.
Much more. It's the origin of bone, cartilage, but also the stem cells for blood cells, the endothelial cells lining blood vessels, and even smooth muscle cells. It's the foundational blueprint for the body's entire supportive and circulatory infrastructure.
Wow. The ultimate progenitor tissue. And that has implications for medicine today, doesn't it?
Huge implications. The exciting thing is, these mesenchyme-like cells, these multipotent stem cells, don't entirely vanish after development. Pockets remain in adults.
Where?
In certain areas of adipose tissue, for instance, and famously in the pulp of your teeth, research is heavily focused on harnessing these adult stem cells for regenerative medicine, coaxing them to repair damaged tissues or even build new organs. It's like trying to reactivate that original embryonic potential.
Incredible potential. Okay, that sets the stage beautifully. Let's move into the matrix itself and meet the residents and the visitors, the cells of connective tissue.
Good way to think about it. We can divide them into two groups: the permanent residents, born and raised locally, and the transient guests, cells that migrate in, usually from the bloodstream, often for defense purposes.
Let's start with the main architects, the builders, the fibroblasts.
Fibroblasts are the principal cells of CT proper. They're permanent residents, derived right from those mesenchymal cells, their big job, to synthesize and maintain almost the entire ECM.
So they're making the collagen, the elastin.
Everything. Collagen, which is, by the way, the most abundant protein in your body, elastin, all the GAGs, the proteoglycans, the multiadhesive glycoproteins, they're powerhouse protein factories.
And you can actually tell if they're active just by looking at them?
You absolutely can. An active fibroblast, one that's busy building, is larger, often has an irregular branched shape because it's pushing out so much material. Inside, you'd see tons of rough endoplasmic reticulum, the protein assembly lines, and a very prominent Golgi apparatus for packaging.
And the nucleus.
Large and euchromatic, just like its mesenchymal ancestor, signaling intense gene transcription. Very active.
What about when things quiet down?
Then you get the quiescent fibrocyte. It's smaller, more spindle-shaped, fewer extensions. Internally, the RER and Golgi are much reduced. The nucleus is smaller, darker, more condensed, heterochromatic. It's essentially dormant, just maintaining the status quo, waiting for a signal.
And what kind of signal wakes it up?
Injury. Exactly. Tissue damage is the main trigger. Growth factors released at the injury site stimulate these fibrocytes to start dividing again, ramp up production, and become active fibroblasts.
This is how scars form, right?
Precisely. Fibroblasts are key to wound healing. They migrate into the damaged area and lay down new ECM. In tissues that don't regenerate well themselves, like heart muscle after a heart attack, fibroblasts fill the gap with scar tissue. It stabilizes the area, but it's not functional tissue.
It's a patch job, essentially.
A vital patch job, but yes.
Yeah.
And sometimes you see a specialized version involved in healing.
The ones that actually pull the wound edges together.
The myofibroblasts. Fascinating cells. They differentiate from fibroblasts but develop contractile proteins, lots of actin and myosin, similar to smooth muscle.
So they can contract.
Yes, they generate force. This is crucial for wound contraction, literally shrinking the size of the defect by pulling the surrounding tissue inward.
Okay, moving from the builders to the defense and cleanup crew. Let's talk macrophages.
Macrophages are defined by their phagocytic prowess. They are the big eaters of the connective tissue.
What are they eating?
Cellular debris, apoptotic cells that are programmed to die, old matrix components like worn-out fibers, bacteria, even potentially cancerous cells. They are essential housekeepers and frontline defenders. You always find loads of them at sites of inflammation.
What do they look like? How would we spot one?
They're quite large, maybe 10 to 30 micrometers across. Their nucleus is usually off to one side, often kidney bean-shaped. But the real giveaway, especially with an electron microscope, is their cell surface.
It's not smooth.
Far from it. It's covered in folds, ruffles, and projections, evidence of constant pinocytosis, cell drinking, and phagocytosis, cell eating. Inside, they're packed with lysosomes, the digestive organelles. Pathologists sometimes call them histiocytes.
Then they come from the blood, right?
Correct. They start life as monocytes, a type of white blood cell circulating in the blood. When they receive chemical signals from a tissue, they squeeze through the walls of small blood vessels, usually venules, enter the CT, and then differentiate into mature, long-lived macrophages.
And this is part of a bigger system, isn't it? The mononuclear phagocyte system.
Yes, the mononuclear phagocyte system, MPS, is this whole network of related phagocytic cells spread throughout the body. It includes the monocytes in blood, the macrophages in CT, but also specialized versions in different organs. Like Kupffer cells in the liver sinusoids, microglial cells in the brain and spinal cord, Langerhans cells in the skin, dendritic cells in lymph nodes, and even osteoclasts in bone, which are giant multinuclear nucleated cells derived from macrophage fusion.
So they adapt to their local environment but share that phagocytic origin. What about their immune role beyond just eating things?
That's absolutely key. They are crucial players in innate immunity, the first line of defense, but they also bridge to adaptive immunity. They act as antigen-presenting cells, APCs.
How does that work?
After they engulf and digest something foreign, like a bacterium, they take small protein fragments, antigens, and display them on their cell surface using special molecules. This presentation is essential for activating T lymphocytes, which orchestrate a much more specific, targeted immune response.
So they're like intelligence agents, showing the specialized forces what the enemy looks like.
That's a great way to put it. And sometimes if the enemy or the debris is too large for one macrophage to handle,
They team up.
they fuse together. They merge their cytoplasm and nuclei to form multinuclear giant cells. You see these often in response to things like splinters, sutures, or certain chronic infections like tuberculosis. It's a sign of a persistent challenge.
Okay, fascinating cells. Next up, the ones responsible for immediate allergic reactions. The mast cells.
Mast cells. Very distinctive. They're large, oval, or irregular cells, maybe 7 to 20 micrometers, and their cytoplasm is just jam-packed with large, dark-staining, basophilic secretory granules.
So many granules you often can't even see the nucleus clearly.
Exactly. They completely obscure it sometimes. And these granules have a unique staining property.
Metachromasia. That always stuck with me from histology labs. Why does it happen?
It's because the granules are loaded with highly acidic sulfated GAGs, particularly heparin. These dense negative charges cause certain basic dyes, like toluidine blue, to stack up in an unusual way, which shifts the color they absorb and emit. So, instead of blue, the granules stain purple or red. It's a chemical signature.
Where do mast cells hang out? Their location seems important.
It's absolutely strategic. They congregate near small blood vessels. We call them perivascular mast cells, and they line the interfaces with the outside world just under the epithelium in the digestive and respiratory tract's mucosal mast cells. They're perfectly positioned as sentinels.
Sentinels waiting to release their cargo. What's in those granules? What's the chemical arsenal?
It's potent stuff designed for immediate local effects. The preformed mediators ready to go include, first, heparin, that anticoagulant JAG. Second, histamine.
The famous one.
Right. Histamine causes blood vessels to become leaky, increasing vascular permeability, and makes smooth muscle contract, like in the airways. Third, serine proteases, enzymes that chop up proteins and activate other inflammatory mediators. And fourth, chemotactic factors that specifically call in reinforcements, like neutrophils and eosinophils.
And they make other things too, don't they? Not just the prepackaged stuff.
Yes, upon activation, they also rapidly synthesize and release signaling molecules like cytokines and potent lipid mediators derived from phospholipids, such as prostaglandins and leukotrienes, which sustain and amplify the inflammatory response.
This rapid release, degranulation, is the basis for things like hay fever and, more severely, anaphylactic shock.
Yeah.
How is that triggered so quickly?
It's a two-step process, essentially. First, exposure to an allergen pollen, bee venom, whatever causes your B lymphocytes to produce a specific class of antibody called IgE.
Yeah.
This IgE doesn't just float around, it binds with very high affinity to special IgE receptors covering the surface of mast cells. Now, the mast cell is sensitized or armed.
Primed for action.
Exactly. The next time you encounter that same specific allergen, it binds to the IgE already sitting on the mast cell surface. If it cross-links two or more adjacent IgE molecules, that's the trigger.
Like bridging them together.
Precisely. That cross-linking event sets off an immediate cascade of signals inside the mast cell phosphorylation, a huge influx of calcium ions, and boom, degranulation. The granules fuse with the cell membrane and dump their entire contents, histamine, heparin, everything into the surrounding tissue almost instantaneously.
Leading to those rapid allergy symptoms. Wow. Okay, let's switch to another key immune player in the CT, the plasma cells.
Plasma cells are the antibody factories. They're terminally differentiated B lymphocytes. Their entire existence, which is relatively short, maybe 10 to 20 days, is dedicated to producing and secreting vast quantities of specific antibodies, or immunoglobulins.
And their appearance really screams factory, doesn't it?
It absolutely does. They're large ovoid cells. Their cytoplasm is intensely basophilic stains, dark blue purple with standard stains because it's crammed wall to wall with rough endoplasmic reticulum for protein synthesis.
So much RER it pushes the nucleus aside.
Often, yes. The nucleus is typically eccentric, pushed to one side, and right next to the nucleus you usually see a pale, unstained region. That's the Golgi apparatus, working overtime to modify, sort, and package the antibodies for secretion.
Hmm.
We call it the juxtanuclear Golgi, or Golgi ghost.
And the nucleus itself has that classic look.
The clock face nucleus. The chromatin, the DNA, and associated proteins tends to clump around the periphery of the nuclear envelope, resembling the numbers on a clock face. It's a very characteristic feature reflecting their specialized state.
And the antibodies they pump out, what's their job?
Their job is neutralization. They bind specifically to antigens like bacterial toxin, think tetanus toxin, or viruses, preventing them from harming cells. These antibody-antigen complexes are then tagged for destruction and rapidly cleared by phagocytes like macrophages.
Okay, so we have fibroblasts, macrophages, mast cells, plasma cells. Any other significant cell populations wandering through?
Yes, the other leukocytes, white blood cells, primarily neutrophils, eosinophils, and lymphocytes that haven't yet differentiated into plasma cells. Like monocytes, they migrate from the blood into the CT, especially during inflammation.
Let's focus on inflammation then. It seems to involve all these players working together. What kicks it off?
Inflammation is the body's coordinated response to injury or infection. It starts when tissue damage or pathogens trigger the release of chemical signals, mediators from damaged cells from the ECM itself, and importantly, from resident sentinel cells like mast cells and macrophages.
These mediators sound the alarm.
They do. They act locally on blood vessels, causing them to dilate and become more permeable. They also activate other nearby cells.
Which leads to those classic signs we all recognize. Redness, swelling, heat, pain. Rubor et tumor cum calore et dolore.
Exactly. The redness, rubor, and heat, calor, come from the increased blood flow due to vasodilation. The swelling tumor is caused by plasma fluid leaking out of those more permeable capillaries into the tissue that's edema.
And the pain.
That comes partly from the pressure of the swelling on nerve endings, and partly from chemical mediators directly stimulating pain receptors. Substances like histamine from mast cells are key players in increasing that vascular permeability, causing the leakiness and swelling.
And inflammation also brings in more troops, right?
Yes, that's crucial. The chemical mediators released also act as chemotactic signals, creating a chemical gradient that actively attracts more leukocytes, especially neutrophils initially, from the blood to the site of injury or infection. It's a targeted recruitment process.
A really dynamic battlefield. Okay, let's shift focus from the cells to physical structures they build and inhabit, the fibers of the ECM. Starting with the undisputed champion, collagen.
Collagen is the star. It makes up about 30% of your body's dry weight. It's the protein that gives tissues incredible strength, resistance to being stretched or torn. It's not just one protein, though. It's a large family, currently 28 known types in vertebrates.
How do we make sense of all those types? Are they grouped somehow?
We mainly classify them based on the structures they form. The most abundant group forms large, visible fibrils, the fibrillar collagens.
That includes the main one, type 1.
Yes, type I is the workhorse, forming thick, strong bundles you find in skin, tendons, ligaments, bone. It resists tension. Type 2 collagen forms finer fibrils and is key for resisting pressure in cartilage. Type 3 forms the delicate reticular fibers. Types V and Ara are usually associated with type I and 2, helping regulate fibril size.
So those are the rope makers. What other forms exist?
There are the network or sheet-forming collagens. The main one here is type 4. It doesn't form fibrils at all. Instead, its molecules assemble into a mesh-like sheet. This is the primary structural component of all basal laminae, the floor that epithelial cells sit on, and external laminae around muscle and nerve cells.
Like a filter or a scaffold.
Exactly. And then you have linking or anchoring collagens. These act like molecular bridges, connecting fibrillar collagens to each other or to other ECM components. Type 7 collagen, for example, forms anchoring fibrils that rivet the basal lamina, made of type 4, to the underlying connective tissue, often containing type I and 3.
Okay, that's a helpful overview. Now, I want to really dive deep, get into the mastery level on how type I collagen is actually made. It's famously complex, a multi-step process inside and outside the cell. 7 steps, right?
At least 7 key stages, yes. It's a remarkable example of protein synthesis, modification, and assembly, orchestrated mainly by fibroblasts. Understanding this pathway is crucial because so many things can go wrong.
Okay, step 1, reading the genetic code.
Transcription and translation. The genes for the collagen alpha chains are transcribed into messenger RNA, which is then translated into polypeptide chains, The procollagen alpha chains on ribosomes attach to the rough endoplasmic reticulum, RER.
What's special about these alpha chains?
They have a very repetitive amino acid sequence. Glycine is found at every third position, GLY-XY, and the X and Y positions are often filled by proline and lysine. This specific sequence is essential for forming the final structure.
Step 2. A critical modification inside the RER.
Hydroxylation. Enzymes within the RER lumen add hydroxyl OH groups to many of those proline and lysine residues. This is absolutely vital for the stability of the final molecule.
And this step needs specific helpers, cofactors.
Typically, yes. The hydroxylase enzymes require molecular oxygen, ferrous iron takes up E2 plus 1O, and ascorbic acid vitamin C to function.
The scurvy connection.
Precisely. If you lack vitamin C, this hydroxylation step fails. Without the hydroxyl groups, the alpha chains can't form the necessary hydrogen bonds to stabilize the triple helix structure later on.
So the collagen produced is defective.
It's unstable. It doesn't fold properly and gets degraded quickly. This leads to the symptoms of scurvy, weak blood vessels causing bleeding gums and hemorrhages, poor wound healing, problems with bone formation, all due to faulty collagen.
A stark reminder of how crucial basic nutrients are. Okay, step 3.
Glycosylation. Still within the RER, specific sugar molecules get attached to some of those newly formed hydroxylysine residues. This also plays a role in guiding helix formation and interactions.
Step 4. Forming the famous triple helix.
Triple helix formation. 3 modified alpha chains align with each other. They're guided into alignment by interactions and disulfide bonds forming at their C-terminal ends, sort of zipping them up from one end. Then they wind around each other into that characteristic right-handed triple helix. This structure is now called procollagen. It's still soluble at this stage.
Step 5. Getting it out of the factory.
Secretion. The folded procollagen molecules are transported through the Golgi apparatus for further modification and packaging, then loaded into secretory vesicles, moved to the cell surface, and released outside the cell by exocytosis.
Now the action moves outside the cell. Step 6.
Extracellular cleavage. Once outside, specific enzymes called procollagen peptidases snip off the loose lobular propeptide ends from both the N and C termini of the procollagen molecule.
Why is that important?
This cleavage converts the soluble procollagen into the much less soluble collagen molecule, sometimes called tropocollagen. Those removed ends were preventing the molecules from spontaneously assembling into larger structures.
And if this step fails, there's a disease associated with that too, right? Ehlers-Danlos type 7.
Yes. If those propeptides aren't cleaved off properly, the collagen molecules can't pack together correctly into fibrils. The resulting connective tissue is weak, leading to hypermobile joints, joints that dislocate easily, and fragile skin, characteristic of certain types of Ehlers-Danlos syndrome.
Okay, final step. Step 7. Building the actual fiber.
Fibril assembly and cross-linking. The cleaved collagen molecules now spontaneously self-assemble end-to-end and side-to-side into larger structures called fibrils. This assembly happens in a specific, staggered way.
A staggered. How so?
Each molecule is offset from its neighbor by about 67 nanometers. This regular staggering creates gaps between the ends of molecules in one row and the molecules in the next. It's this pattern of gaps and overlaps that produces the characteristic cross-striations or banding pattern you see on collagen fibrils in electron micrographs.
So the pattern reflects the molecular arrangement. Is that assembly strong enough on its own?
It's organized, but it needs reinforcement. The final touch is covalent cross-linking. An enzyme called lysyl oxidase, also acting outside the cell, creates strong covalent bonds between adjacent collagen molecules within the fibril, locking them together. This cross-linking is what gives collagen its immense tensile strength.
An incredible process. But collagen doesn't last forever, does it?
Yeah.
It needs to be broken down and remodeled.
Absolutely. Tissue remodeling, wound healing, even normal growth requires collagen degradation. This is initiated by specific enzymes called collagenases.
Who makes those?
They're part of a larger family called matrix metalloproteinases, or MMPs, and they're secreted mainly by macrophages, but also fibroblasts and other cells. These collagenases make specific cuts in the intact collagen triple helix, which then allows other, less specific proteases to finish the job of breaking it down completely.
Controlled demolition process. Okay, let's move to the next fiber type. Reticular fibers. These are more delicate.
Much more delicate. They're composed mainly of type 3 collagen. Instead of forming thick bundles like type A, type 3 forms very thin fibrils, 0.5 to 2 nm diameter that branch and interconnect to create a fine mesh-like network or reticulum.
Are they easy to see in standard tissue preparations?
No, they're usually invisible with regular H&E staining. You need special stains to visualize them.
Like silver stains.
Exactly. They are argyrophilic, meaning they love silver and stain black with silver salt impregnation methods. They are also PAS positive.
PAS positive. What does that mean chemically?
Periodic acid shift stain positive. It indicates the presence of abundant carbohydrates. Reticular fibers have a much higher sugar content, maybe up to 10% attached to the type 3 collagen, compared to the big type I fibers, which have only about 1%. That's why they stain with PAS.
And where do we find this delicate supportive meshwork? What's its function?
It forms the supporting stroma, or framework, for organs with lots of free cells or glands, places where the tissue architecture needs to be flexible but supportive. Think of the liver, bone marrow, lymph nodes, spleen, endocrine glands. It provides a scaffold for the functional cells to sit within or migrate through.
Supporting rapidly changing cell populations. Makes sense. Okay, the third major fiber type, elastic fibers and lamellae, the ones that stretch.
Right. These provide the rubber band quality to tissues. They allow tissues like the skin, lungs, and especially large arteries like the aorta to stretch significantly under pressure and then snap back to their original shape when the pressure is released.
In arteries, they're organized into sheets, aren't they?
Yes, in the walls of large arteries, you find concentric layers of elastic material called elastic lamellae, which are basically fenestrated sheets, like sheets of Swiss cheese, allowing cells and fluids to move through.
What are they made of? It's not just one protein, is it?
No, it's a composite structure. First, a scaffold of thin microfibrils is laid down, made primarily of a glycoprotein called fibrillin. Then, the main component, the protein elastin, is deposited onto this fibrillin scaffold. Elastin makes up the bulk of the mature elastic fiber.
How does elastin provide that rubber-like elasticity? What's the molecular basis?
Elastin molecules are rich in hydrophobic amino acids and normally exist in a kind of random coil state like a tangled string. When you stretch the fiber, these molecules get pulled into a more linear arrangement.
But what holds them together so they don't just pull apart?
That's the key. Unique crosslinks. Elastin molecules are covalently linked together by very unusual structures called desmosine rings. These are formed from 4 lysine residues on adjacent elastin molecules that get chemically modified and joined together by lysyl oxidase, the same enzyme involved in collagen crosslinking.
So these desmosine crosslinks act like permanent ties between the elastin chain.
Exactly. They hold the whole network together, but still allow the individual molecules to stretch and relax using hydrophobic interactions to drive the recoil. It's this cross-linked random coil structure that gives elastic fibers their remarkable properties.
And if that scaffolding protein, fibrillin, is defective, we mentioned Marfan syndrome.
Yes, mutations in the fibrillin 1 gene cause Marfan syndrome. Because fibrillin forms the essential scaffold, if it's faulty, the elastin doesn't get organized properly. The resulting elastic tissues are weak and lack resilience.
Which is particularly dangerous where?
In the aorta. The constant high pressure from the heartbeat stresses the weakened aortic wall, leading to a high risk of aortic aneurysm, a dangerous bulge, or dissection, a tear in the wall, which can be fatal. It highlights how critical that fibrillin-elastin interaction is.
A sobering example. Okay, we've covered the fibers in detail. Let's move to the other major ECM component, the ground substance, the hydrated gel.
Right, this is the amorphous, transparent material filling all the spaces between the cells and fibers. It's highly hydrated, viscous, and acts as both a lubricant and, importantly, a physical barrier to the penetration of microorganisms. Plus, it's the medium for diffusion.
Let's break down its molecular components, starting with the glycosaminoglycans, GGs, the big water binders.
GGs are long, unbranched polysaccharide chains made of repeating disaccharide units. One sugar is usually a uronic acid, the other an amino sugar. They are intensely negatively charged due to sulfate and carboxyl groups.
And that negative charge is key.
Absolutely. The charges cause the GAG chains to repel each other and attract huge amounts of water and positive ions, like sodium, forcing them into an extended conformation that occupies a large volume. This creates the swelling pressure, or turgor, that helps tissues resist compression.
Is hyaluronic acid the biggest GAG?
By far. It's enormous, thousands of disaccharide repeats, reaching molecular weights in the millions. And it's unique in another way, too.
How so?
Unlike the other GAGs, which are made inside the cell, in the Golgi, and attached to proteins, hyaluronin is synthesized directly at the cell surface by an enzyme complex called hyaluronin synthase and extruded directly into the ECM. It forms massive viscous networks that are great for lubrication, like in joints, and facilitating cell migration.
What about the other GAGs, the sulfated ones?
These are smaller chondroitin sulfate, dermaton sulfate, heparin sulfate, keratin sulfate. They are synthesized inside the cell, sulfated to varying degrees, and covalently linked to core proteins to form the proteoglycans we'll discuss next. Their high density of negative charge makes them crucial for binding water and contributing to the gel state.
And problems breaking these down lead to diseases.
Yes, the mucopolysaccharidoses. These are genetic lysosomal storage diseases where individuals lack one of the specific lysosomal enzymes needed to degrade a particular GAG. The undegraded GAGs accumulate within lysosomes, disrupting cell function and causing progressive damage to multiple organs. Examples include Hurler, Hunter, and Sanfilippo syndromes.
A tragic consequence of a single enzyme failure.
Okay.
So these sulfated GAGs are usually attached to proteins, forming proteoglycans, PGs.
That's right. A proteoglycan consists of a central core protein synthesized in the RER, to which multiple GAG chains are covalently attached in the Golgi. Often, the total mass of the GAG chains far outweighs the mass of the core protein itself. Think of them like bottle brushes, with the protein as the wire core and the GAGs as the bristles.
And these bottle brushes don't just float around individually?
Often, no. Many proteoglycans, especially in cartilage, assemble into even larger structures. Numerous PG molecules, like aggrecan, can bind non-covalently via special link proteins to a single long backbone of hyaluronin,
Creating a massive complex.
a proteoglycan megacomplex. These structures are gigantic and incredibly efficient at trapping water, making tissues like cartilage incredibly resilient to compression.
Could you give examples of specific proteoglycans and their roles?
Sure, aggrecan is the major one in cartilage, packed with chondroitin and keratin sulfate, responsible for its shock-absorbing properties. Perlecan is a key proteoglycan found in all basal laminae. The corin is smaller and binds to the surface of type I collagen fibrils, helping to regulate their assembly and spacing. Syndecan is interesting because its core protein actually spans the cell membrane, linking the ECM directly to the cell's internal cytoskeleton.
So they're structural, but do they do anything else? You mentioned signaling before.
Yes, that's a crucial, perhaps less obvious function. PGs can bind and sequester various signaling molecules, including growth factors like FGF, fibroblast growth factor, within the ECM. They act like reservoirs.
Holding on to them.
Exactly. Then, when the ECM is partially degraded during tissue injury or remodeling, these trapped growth factors are released locally, right where they're needed, to stimulate cell proliferation and repair. It's a clever way to regulate signaling.
Very clever. Okay, the last component of the ground substance. The multiadhesive glycoproteins. The molecular glue.
Right. These are large glycoproteins with branched sugar chains and, critically, multiple binding domains. They act as connectors, linking cells to the ECM and linking different ECM components together.
Who are the main players here?
Two big ones are laminin and fibronectin. Laminin is primarily found in basal laminae. It's a large, cross-shaped molecule with binding sites for cell surface receptors like integrins, type III collagen, and proteoglycans. It's essential for organizing the basal lamina structure and mediating cell attachment to it.
And fibronectin.
Fibronectin is more widespread, found throughout connective tissues. It's a dimer produced mainly by fibroblasts. It binds to various types of collagen, GAGs, and cell surface integrins. It's crucial not just for cell adhesion, but also for guiding cell migration during development, and wound-healing cells literally crawl along fibronectin pathways.
How do cells actually grab onto these laminins and fibronectins?
Through cell surface receptors called integrins. These are transmembrane proteins. They span the cell membrane. Their extracellular part binds specifically to sequences on fibronectin, laminin, or collagens, while their intracellular part connects to the actin cytoskeleton inside the cell.
So they link the outside environment to the cell's internal machinery.
Precisely. And interestingly, integrins typically bind to their ECM ligands with relatively low affinity.
Low affinity? Wouldn't high affinity be better for strong attachment?
You'd think so, but the low affinity allows for dynamic interactions. Cells can easily make and break connections as they move, probe their environment, or respond to signals without getting permanently stuck. It allows for flexibility.
And when many integrins cluster together,
They can form structures called focal adhesions. These are concentrated patches where integrins link the ECM outside to bundles of actin filaments, stress fibers, inside the cell, often via adapter proteins like talanin and alpha-actinin.
What's the significance of these focal adhesions?
They're not just anchors, they're mechanosensors. They allow the cell to feel the physical properties of the ECM, its stiffness, tension, and translate those physical cues into biochemical signals inside the cell, influencing things like cell shape, migration, proliferation, and even gene expression. It's how cells read their physical environment.
Amazing. The ECM is truly an active, information-rich environment. Okay, section 5. Let's talk about the fluid moving through this matrix, interstitial fluid dynamics.
Right, the fluid bathing the cells and fibers is the interstitial fluid. Its basic ion composition is similar to blood plasma, but it has much lower protein concentration, although some plasma proteins do leak out of capillaries. In fact, because CT is so voluminous, a significant fraction, maybe up to a third, of the body's total plasma protein is actually found in this interstitial space at any given time.
And if too much fluid accumulates here, that's edema.
Exactly. Edema is the excess buildup of interstitial fluid. It happens when the balance between fluid leaving the capillaries and fluid returning is disrupted. This is common in inflammation, where inflammatory mediators make capillaries much leakier, allowing excess fluid and protein to escape into the tissue.
What normally drives fluid movement across capillary walls? It's a balance of pressures, isn't it?
It is. Two main opposing forces are at play, often called Starling forces. First, hydrostatic pressure. This is the physical pressure of the blood being pushed through the capillary by the heart. It's higher at the arterial end of the capillary and tends to force water out of the vessel into the interstitium.
Okay, pressure pushing out. What pulls it back in?
Call it osmotic pressure or oncotic pressure. This is generated by the plasma proteins, mainly albumin, that are trapped inside the capillary because they're too large to easily cross the wall. Their high concentration inside draws water back into the capillary by osmosis. This force is pretty constant along the capillary, but becomes dominant at the venous end, where the hydrostatic pressure has dropped significantly.
So fluid gets pushed out at the start, and most gets pulled back in at the end. What happens to the little bit left over?
That small amount of excess fluid, along with any leaked plasma proteins, is continuously drained away by a separate network of vessels, the lymphatic capillaries.
Ah, the lymphatic system.
Right. These tiny, blind-ended lymphatic vessels collect the excess interstitial fluid, now called lymph, and eventually return it back to the bloodstream, preventing edema and also allowing immune cells to monitor the fluid for antigens.
A critical drainage and surveillance system. Okay, finally, let's wrap up with the broad classification of connective tissue proper. How do we categorize the main types?
It's primarily based on the relative amounts and arrangement of the components, especially the collagen fibers versus the ground substance. We broadly divide it into loose and dense CT.
Let's start with loose connective tissue, sometimes called areolar tissue.
Yes. This is characterized by having roughly equal parts of cells, fibers, and ground substance. It has quite a lot of viscous ground substance. All the resident and transient cell types we discussed are typically found here, though fibroblasts are usually the most numerous. Collagen fibers are present, but they're relatively sparse and interwoven with elastic and reticular fibers.
What are its properties? Where is it found?
It's delicate, flexible, well vascularized, but not very resistant to stress. It's very common, typically found filling spaces between organs, supporting epithelia as the lamina propria beneath mucous membranes, surrounding blood vessels and nerves. It's like the body's packing material.
Okay, then we have the dense types. First, dense, irregular connective tissue. What makes it dense and irregular?
Dense because collagen fibers clearly predominate over ground substance and cells. There are far fewer cells, mostly fibroblasts. Irregular because the collagen fibers, mainly thick type I bundles, are arranged randomly, interwoven in a meshwork going in all directions.
What's the functional advantage of that random arrangement?
It provides significant strength and resistance to stresses pulling from multiple directions. Think about the dermis of the skin it needs to resist, stretching every which way. You also find dense, irregular CT forming tough capsules around organs like the testes, liver, and lymph nodes, and in the submucosa of the digestive tract.
It's the tough protective layer. Now, the other dense type. Dense, regular connective tissue.
Here, dense again means packed with type I collagen fibers, very little ground substance, few cells, mostly elongated fibroblasts called tendinocytes, and importantly, few blood vessels. Regular means the collagen bundles are arranged in parallel alignment, all oriented in the same direction.
That parallel structure dictates its function.
Absolutely. It provides tremendous resistance to tension and pulling forces exerted consistently along one axis. This is the tissue that makes up tendons, connecting muscle to bone, and ligaments, connecting bone to bone, where forces are primarily unidirectional. Aponeuroses, which are sheet-like tendons, are also dense regular CT.
You mentioned few blood vessels. Does that impact healing?
Greatly. Because of the poor blood supply, tendons and ligaments heal very slowly after injury. Inflammation, like tendinitis from overuse, can be persistent and difficult to resolve because repair cells and nutrients have limited access. The fibroblasts have to slowly remodel that very dense matrix.
Makes sense. Are there any other specialized CT types we should briefly mention?
2 more were noted in the sources. Reticular tissue, which we touched on earlier, it's characterized by a meshwork of fine reticular fibers, type 3 collagen, forming the supportive stroma in lymphoid organs like the spleen, lymph nodes, and bone marrow, supporting the blood-forming and immune cells.
And the one found in the umbilical cord?
Mucoid tissue, or mucous connective tissue. This is the main component of the fetal umbilical cord, known as Wharton's jelly. It's very gelatinous, with abundant ground substance, rich in hyaluronin, but sparse collagen fibers and scattered, often stellate-shaped fibroblasts. It's interesting because it's rich in those mesenchymal stem cells we discussed at the beginning. You also find similar tissue in the vitreous humor of the eye and the pulp of young teeth.
Right, Wharton's jelly. Okay, that covers the classification. We've gone incredibly deep into the components, cells, fibers, ground substance, dynamics, and types. Let's try to synthesize the key takeaways for someone aiming for mastery here.
I think the core concept is that connective tissue is the extracellular matrix. Understanding the ECM's composition and how its components interact is key.
We saw that synergy: collagen for tensile strength, elastin for recoil, and the hydrated GAGs and proteoglycans in the ground substance, creating that essential environment for cushioning, diffusion, and even regulating cell behavior by binding growth factors.
Exactly. And then understanding the cellular players, the resident builders, the fibroblasts constantly maintaining the matrix and capable of ramping up for repair, and the crucial immune sentinels and responders, macrophages for cleanup and antigen presentation, mast cells for triggering immediate inflammatory responses.
We also saw the clinical relevance woven throughout how the complex collagen synthesis pathway depends on things like vitamin C, and how genetic defects in ECM components lead to systemic diseases like scurvy, Ehlers-Danlos, and Marfan syndrome.
It really highlights how interconnected structure and function are at the molecular level.
It's an incredibly dynamic system, isn't it?
Yeah.
Constantly being monitored, remodeled, defended.
Which leads perfectly into a final thought, perhaps. We talked quite a bit about the mononuclear phagocyte system and how incredibly adaptable these cells are. A monocyte from the blood becomes a tissue macrophage. That macrophage can become highly activated, it can fuse with others to become a multinuclear giant cell to tackle large debris, or it can even differentiate further into a specialized cell, like an osteoclast that dissolves bone.
That plasticity is remarkable.
It really is. So the provocative thought is this. Given this inherent capacity for cells within the connective tissue environment to completely change their identity and function, literally swap jobs based on the signals they receive, what does this plasticity tell us about the body's ongoing negotiation with injury, debris, and disease? And more importantly, what's the ultimate therapeutic potential if we could learn to precisely control those molecular switches? Could we intentionally guide these cellular transformations towards regeneration in tissues that currently scar, or could we prevent harmful transformations in chronic disease?
The potential to harness that inherent cellular adaptability, that's a profound place to leave things. A fantastic deep dive into the matrix. Thank you.
It was my pleasure. It's a fascinating and fundamental topic.